All right, let's get started here. Welcome, everybody. This is the final week in the GCOOS Spring Series. I'm Krista Manello. I'm the Outreach and Education Manager for GCOOS. I'll be moderating today. Jen Vreeland is also helping today. If you have any questions during the presentations or after, you can use the chat box, or we're a small enough group. Feel free to just raise your hand and we'll have an informal conversation that way. We'll have two presentations today. First will be Dr. Lisa Campbell, and then following her talk will be Dr. Kenneth Hoadley. I will introduce Lisa first. We'll have time for questions, and then I'll introduce Dr. Hoadley after that. So Dr. Lisa Campbell, welcome. She's the Regents Professor and William Bryant Endowed Chair in the Department of Oceanography at Texas A&M University College Station, where she also has a joint appointment in the Department of Biology. Her research is focused on the diversity and population dynamics of phytoplankton, especially HABs in the Gulf of Mexico. She's been a pioneer in the use of the imaging flow cytobot, also known as the IFCB, that combines flow cytometry and video technology to monitor harmful algae in real time. And since 2007, the IFCB has been the backbone of the Texas Observatory for Algal Succession Time Series, also known as TOAST, allowing for early warning of HAB events with sufficient time to close shellfish harvesting. If needed, Lisa is not just a pioneering researcher. She's been recognized for distinguished teaching and outstanding mentoring. She served on the board of directors of ASLO and is an associate editor for the Journal of Plankton Research. And her talk today is titled Early Warning of Harmful Algal Blooms Using Continuous Automated Imaging Flow Cytometry. So following your talk, Lisa, we'll have time for questions. So all yours. Thanks.
Great. So I will share. I'm sharing my screen. Oops. So does everyone see my screen?
Yes, it looks great.
Yes. Okay. So thank you very much for the invitation. I'm going to be telling you about our use of the imaging flow cytobot that Darren and I have done. Dr. Darren Henricks is an assistant professor, instructional professor in the department, along with me. We've been working on this since 2008. Next. Oops. It's not advancing. Oops. Okay. So the outline of this presentation, I'll first introduce HABs on the Texas coast, although most of you are probably familiar, and then what the requirements are for early warning, and then a little bit about the instrument itself, both the operation and then our deployments at TOAST, and then tell you about the successful early warnings that we've had for both Dinophysis ovum, which can you see my cursor here? And then Caranea brevis. And then some of the issues that we've addressed over the years and how we've developed an automated alert system for the early warning of HABs. So, and then following this, I'll mention some of the challenges to the operations and some of the future observing capabilities that we'd like to develop. And then finally close with how we align with the GCOOS strategic plan. So, as you know, harmful algal blooms are principally the red tide Caranea brevis, which is shown here in a bloom in South Padre Island, and the principal impacts then are killing fish here piled up on the beach, and then the route of exposure for humans is principally through eating contaminated shellfish, causing neurotoxic shellfish poisoning, or through inhalation of the aerosols that we heard last week from Barb Kirkpatrick. But there are also additional harmful algal species, and more principally Dinophysis ovum, which blooms in the spring, whereas Caranea brevis tends to bloom in the fall months. And then a recent identification of an additional species that does produce some toxins, although we have not had any human impacts from this one reported as of now. But really, since we do not know how to predict when and where blooms occur, the early warning is the most effective mitigation for harmful algal blooms. So the requirements, though, for an effective early warning system is regular sampling, high frequency, and high accuracy. So the Cytobot, the imaging flow Cytobot, is really the best instrument to accomplish these goals. So it was designed originally by Rob Olson and Heidi Sosik at the Woods Hole Oceanographic Institution, and this is the publication that they published in 2007 on this, the original instrument. But now it's, of course, commercially available from McLean Research Laboratories. Ivory Eggstrom built the first one that we had in our lab, and they now have a team of engineers that are producing them. And so as you all probably know, that this instrument combines, this shows here, the out of the can, it combines flow cytometry and video technology to image the phytoplankton. And it is submersible, and it allows for unattended continuous operation. So that really is important for HAB detection. So for those of you not familiar with the instrument, I'll just show briefly. It combines flow cytometry. So here's a laser, and cells pass the laser in single file, and then the fluorescence is detected by detectors here. And then if there's a sufficient chlorophyll fluorescence that due to the pigment in the cells, the phytoplankton cells, then a CCD camera is triggered. And this there. And then this is the flow cell that focuses on the particles as they're going by in single file. And that's the flow cytometry aspect of this, is that individual particles can be imaged. And this is a image lower here of the syringe that takes a 5 ml sample every 20 minutes, and so we get about 70 samples a day. And for anybody who's done phytoplankton counts, you know that this saves a lot of time for having it automated. And so also this is it can operate 24 hours, 7 days a week, which a person just cannot do. So once the data are imaged and stored on the computer of the instrument, then the data are copied to my lab here in College Station, where they're archived and then processed further and then we can generate an automated notification if there are sufficiently numbers of the harmful species, which I'll show in a few slides. So the network of IFCVs that we've deployed, we first started at Port Aransas on this pier at the time in 2007, and then in 2017 we deployed a second instrument at Surfside Beach, which is near Freeport, Texas, and this was deployed. The Port Aransas instrument was deployed in the pier lab, so it wasn't submerged, but in Surfside Beach we did deploy it in combination with a sonde, submersed, and this then has become the backbone for the Texas Observatory for Algal Succession Time Series, or TOAST. It took us a long time to come up with that acronym, but as you know, in oceanography, acronyms are everything. So here's a picture of how it's deployed, or was deployed. The pier, as you may know, was destroyed after Hurricane Harvey, and it's been rebuilt, and we plan to redeploy this instrument on the pier next week. Then this is the deployment at Surfside. This is the former NOAA weather platform, and we've added a deck, and then we've deployed the Cytobot as a submerged operating instrument. And due to a storm, we now have the power, though, up on top here, because everything was submerged last year during, I can't remember which storm, Ivan or Ian, or what. We've had so many lately, I can't remember which one. But we've had to move the power supply up above, and I'll talk more about that later. So how has this system been used for early warning? Luckily for us, but maybe not for Texas Coast, in its first year of operation, we noticed early on in February a increased number in this species, Dinophysis, and then by the beginning of March, blooms. So we noticed it here first, then we collected a sample towards the end, and water samples were sent to the FDA lab, and then it was okadaic acid was detected. And then in the 1st of March, we collected oysters, and they were found to have 2 to 3 times the recommended level for oyster closures. And then a week later, a recall was issued, and that coincided, of course, with the Fulton Oyster Festival, where 20,000 people attend and have oyster eating contests. So this was a very lucky find that we could prevent Texas oysters from being used. So they recalled all the Texas oysters and used Louisiana oysters at the festival instead. And therefore, a lot of people were saved from getting ill. And another advantage of the Cytobot, of course, is that we can also detect then when the blooms dissipate too, shown here a month later. And so having this abundance at this figure shows at 2-hour resolution. It just shows how patchy things are. But at some instances, conventional microscopy samples did not detect any samples, whereas the imaging flow Cytobot, because of the continuous operation, did detect cells in the water. So since that bloom, since 2008, the IFCB has provided successful early warning for 7 blooms of Dinophysis and or shellfish closures that have resulted in. So you can see, though, not every year a bloom occurs, but they have occurred frequently enough that this has proved very effective for early warning. And we've noticed over the years that the timing or the phenology of blooms occur at different times. So in 2008, it was in mid to late February, but then in 2010, the bloom didn't occur until much later in the year. And over the years, we've noticed considerable timing, and it seems to be related to the temperature. When the water warms up is when the blooms tend to occur. And these data, and plus we isolated Dinophysis ovum for the first time, and we're able to do experiments with it. And these have formed the basis of a model that we hope to use to predict when Dinophysis does occur. And this work is in progress by my former grad student, Jim Ferrandino. In terms of Karenia brevis, we also have had success. This allows us to, again, detect the blooms, in this case, about a month before the peak blooms occur, when dead fish start piling up on the beach. And I just want to point out that the state of Texas does not have a routine monitoring program. They just typically wait till people report a bloom or dead fish start piling up. So having this early warning has been very effective. It's species specific because of the imaging capability, and the peaks of abundance, again, may be missed if by routine monitoring, because routine monitoring doesn't start until blooms are seen. And then also just notice that in this time series, you can see that the blooms are extremely patchy. So we noticed that at this time, the blooms typically showed increasing counts on incoming tides, which suggested that the blooms were coming from offshore. This is the same observation we noticed for the Dinophysis blooms previously. So early warning also provides the Department of Health and the management agencies the time to set up shellfish sampling and testing statewide monitoring more efficiently, so they're not just sampling blindly, and they can focus their efforts to when we first start detecting blooms, and then can focus their sampling of the oyster tissues and then determine the time that's appropriate for closure. So in the last 15 years, we probably have over 1 billion images, and of course that's not possible to manually count. So we need to have some sort of automated classification, and in fact this is essential to have timely analysis. So this data analysis we have developed over the years some training sets. These are manually classified, so we look through the images and pull out images of each of a number of different strains, not just the harmful species, but other species as well. And the initial approach was used based on Heidi Sossik's program using a random forest or a decision tree process, and we used 300 to 500 images in each category. But currently then, Darren Henrichs, my colleague, has used a different approach called a convolutional neural network, and we have over 100 categories now, and we have at least 1,000 images for each of these categories that have allowed us to improve our capabilities of our autonomous automatic classification program. And one of the improvements that is relevant for our HAB work is that Dinophysis and Porocentrum, these two dinoflagellates, do look fairly similar, and so they do tend to occur in the same period, in the February through April time period. And so because of this, there can be some false positives where Porocentrum is identified as Dinophysis. But the CNN classifier is able to distinguish these two species much more clearly, so that's been a vast improvement of our analysis protocol. So another improvement is that Darren has contributed to is eliminating MATLAB, which is a costly program, and we now have a process that just takes the data and it uses just Python, which is a freeware or shareware program, and then feeds that through our automated image classification for the HAB taxa, and then and so we do not require MATLAB for our processing of our data. I failed to mention earlier on, so that we download the data from the field to my lab every hour and processing, so the data are available within about 2 hours of acquisition. So in terms of the automated messaging to our stakeholders, we wanted to devise a threshold for when we make the report. We don't want to have too many false positives or bombard Kirk Wiles, who's our colleague at the Department of State Health Services here in Texas, with too many messages. And so here is a time series from the bloom in 2009, and where we determined that since 5 cells per mil is the legal limit when they close the oyster harvesting, we thought 2 was an effective threshold, because that this would avoid too many false positives, but still provide a month or several weeks at least early warning of when the bloom was just getting started. And so that's the threshold we now use. So when cells are greater than, from our automated classification system, we do send automated emails to my lab and to local state managers in the Texas Department of Health Services, State Department of Health Services, and the Texas Parks and Wildlife. And this is just an example of an email message that they get the last hourly counts for the previous 10 hours. And in this case, this was a bloom of Corenia that was developing, and then they can go to the dashboard and see the images and say, yes, in fact, this is a Corenia brevis cell. So and of course all the state managers are very familiar with what these cells look like, so they can confirm that it's not a false positive. So with this system, we've now had 9 early warnings for HAB events and for either Dinophysis in the spring or Corenia in the fall, and there have been no NSP, neurotoxic shellfish poisoning, or diuretic shellfish poisoning illnesses that have been reported in this time period. So this, we feel, is a good success story. If you drill down into the GCOOS website, you can find these data, although they have not been updating, and we're working on updating the website. I hope this will be done soon. So as I mentioned, there are challenges with this program. Biofouling, we have to clean it, obviously, but we do have programmed daily cleaning for the tubing inside, and we have copper mesh and try to avoid as much fouling as possible. But that is always an issue. Power, as I mentioned, right now it requires to have some plugged into a power source. And so this is our air-conditioned unit utility box that we had on the dock at Surfside, but now, as I mentioned, it's up on top so it doesn't get flooded out. But there are other options such as fuel cells or solar power options that other people have used. We've not gone that way because we have shore-based power available at our locations. We also need an internet to be able to download the data, and so we've used a hotspot or a cell phone for this purpose, although if you're at a university, you could, with the internet availability, you can use local internet. You do need a secure site. We've had issues, of course. A government, the Surfside is a military, a Coast Guard station, so that's very secure, and UTMSI has become more secure. However, you can't prevent everything that may happen, and if anyone wants further information, I can discuss this later. So in summary, TOAST, a network of Cytobots, has been operating in Texas since 2007. We have a continuous time series of the phytoplankton dynamics, and have provided an effective alert system using automated emails to the state managers. And we've had, with work with convolutional neural networks, we have improved image analysis and classification for HAB taxa. And so that the very high temporal resolution has provided 9 successful early warnings, and our current analysis pipeline does not require MATLAB, so that's a cost savings for end users. In terms of aligning with the strategic plan, we align with the issue of developing or preventing or identifying coastal hazards and healthy ecosystems and living resources. And the #1 goal of the human and health safety area of the focus is to improve reporting of harmful algal blooms, and I think the Cytobot does this very effectively. And then we also contribute to all 4 of these cross-cutting themes, and we hope to contribute more to the numerical modeling and forecasting in future. So in the future, we as I said, we have Port Aransas and Surfside, and we hope to have Chip Ryer's instrument down in South Padre added to the network for Texas. And then in terms of networking, of course, as our next speaker will tell you about his deployments in Alabama, and then I know Florida now has them, so we do have a much improved coverage in the Gulf of Mexico that is added to. Of course, the original deployment was Heidi Sossik's and Rob Olson's on Martha's Vineyard, and there's now operations on the East Coast and the West Coast, and of course this will only improve as we do have more and more data. So, and this is the link that you can see the time series at Port Aransas, and then you can also pull up the time series for Surfside at that location as well. So in conclusion, then we've established a HAB network. We have a CNN that's improved HAB identification from the Cytobot images. We have an automated alert system, and our future goals are to continue the time series to evaluate community dynamics, and we're also continuing to develop more additional methods such as metatranscriptomes analysis that would be queued by the time series to help predict the conditions under which harmful algal blooms do bloom. And so I think we are really aligned with the strategic plan in that we maintain what the GCOOS funding is used for, maintains the Cytobot, and for also our data is used in ground truthing the NOAA's HAB bulletin. And then I've already mentioned the strategic plan alignments, and in particular, in particular the human health and safety, and that we are identifying and improving the detection of HABs in coastal waters. And then how does this advance observing and monitoring capabilities? Again, the continuous and automated operation is really what makes this successful, and for early warning and for to obtain data for human health and saving, and for data modeling, and for retrospective analysis of bloom initiation, that we can hope to improve models for in the future to predict harmful algal blooms. Also, of course, since all the data are archived, we can do a lot of additional analyses of phytoplankton community structure and diversity, which feeds into the work that Frank Mueller-Carger presented a few weeks ago. So with that, I'd like to acknowledge all my colleagues, and also the funding agencies that have funded this work. So thank you very much.
Thank you so much, Lisa. That was great. It's always inspiring to hear you've reached the holy grail, right? Everybody wants their information to be used routinely in decision making, and that's certainly where you are with IFCB for TOAST. I'd like to open it up to questions. You can put them in the chat box. You can raise your hand. You can come off camera if you'd like. Turn your microphone on. Kenny, please.
Thank you. Really cool talk, Lisa. I was particularly interested with some of the future directions that you mentioned at the end, the metatranscriptomics.
Yeah.
Is this a device you're developing so that it automatically takes a sample when sort of a threshold is breached? In terms of...
No, unfortunately, not yet. Although I had already spoken with Chris Scholin a long time ago about maybe coordinating the ESP with this, but which Mamari has done. I guess there have been some, but no. Right now, we just manually collect samples when we go for routine analysis down. And we've been actually doing mostly metabarcoding initially. We've been doing that weekly sampling. So okay. It would be great to implement. And people always ask, do we archive samples throughout? And no, we don't do that. Just we don't have storage capability. But that would be another, because sometimes you want to go back and say pick cells and do to do DNA analysis or metabarcoding of the samples. But we have not yet employed that. But that would be something certainly that would be interesting to add.
Gotcha. Thanks.
Thanks, Kenny. Joe, go ahead.
Yeah, Lisa, great presentation. As a board member, it's very reassuring to see that our investment is really making a difference. It's going to good purposes. My question is a basic one. What's the current cost of the Cytobot?
The cost is $135,000. But if you pay a person and their fringe benefits and all that, I mean, it's going to get up there. So in 2 years, and then one person can't count the number of samples that this instrument can do. Now but then you do have to have someone. Oh, and I should also mention, I used an older slide for my acknowledgments. Tomo Sari, who's now working for GCOOS, was also an important member of keeping this system going. You need someone to keep it going because of the fouling and just all sorts of things can go wrong and have gone wrong. So you do need someone to keep it going.
Good. Thank you.
And he's been very helpful. So, but
Lisa, we've also got a couple.
Okay.
Oh, go ahead. Sorry. Nope.
I was just going to say the cost shouldn't be a deterrent, although it is for a number of city and state agencies. Hopefully, for GCOOS, it shouldn't.
We do have a couple questions in the chat box that I'll read for you.
Okay.
From Raul, you have a great presentation. Do you have any experience using IFCBs in freshwater systems?
No, but there are people who have used them, and they've used in the Baltic, which is pretty fresh, 5 parts per thousand. And you just would need to, of course, develop the training sets because you'll have different phytoplankton there. The problem also with a lot of freshwater habs, of course, is that they're filamentous or clumpy, like microcystis things. And so those aren't amenable to a flow cytometer. If the cells that are clumpy or long chains will clog the flow system, and so that's not useful or
Thanks.
possible.
We've got a question from Brock, who would like to know if you're using other technologies for monitoring, such as chlorophyll A sensors, dissolved oxygen sensors, etc., for early warning systems, and why or why not?
Well, we do have a fluorometer on the sonde that we use just for total biomass kind of but all phytoplankton have chlorophyll, so it's not really useful to use just chlorophyll as a biomass indicator for HABs. I think the images have been really the best and most, because again, you need species-specific identifications. So that's so I haven't used any other. I guess toxins would be the other assay. Say, especially in freshwaters, toxins are really important. And we do always, once we detect something, the threshold, the emails go to Kirk Wiles and their whole shellfish sanitation program, and they go out and sample oysters, which of course is the means of preventing human exposure to the toxins. Does that answer your question?
Brock, do you want to acknowledge in the chat? Does that?
Yeah, he did say. He did
okay.
say.
Okay, great. I still see, Joe, your hand is still up. Did you have another question? Okay, great.
All right.
Thanks so much, Lisa, and thanks to everybody for the great questions. Next up, we have Dr. Kenneth Hoadley. Kenny's an assistant professor in the Biological Sciences Department at the University of Alabama and a senior marine scientist at the Dauphin Island Sea Lab. Before taking on these roles, he conducted postdoctoral research at both MBARI and the Geomar Helmholtz Center for Ocean Research in Kiel, Germany. His background is in algal ecophysiology and response to climate change, and encompasses molecular and physiological characterization, including development of custom bio-optical tools. He's part of a collaborative that is working to improve detection and forecasting of harmful algal bloom species in Mobile Bay, where, like Lisa, he's also using the IFCB. He's partnering with GCOOS to create a new imaging analysis pipeline for real-time monitoring of HAB species using the GCOOS data portal. And his talk today is titled Building Capacity for Real-Time Monitoring of HAB Species Within the Gulf of Mexico. Thanks for being here, Kenny. All yours.
Thank you very much, and thank you for that nice introduction. It's been a lot of fun listening to the webinar series through YouTube. So thank you, GCOOS, for making that available on YouTube. We've learned a lot about the different capabilities through GCOOS and all the exciting work that's being done in the area. I am very new to ocean observing and harmful algal bloom research as a whole. So I thought I would start with a brief background on myself. Most of the research I've done thus far has been on the dinoflagellates that live in symbiosis in the host tissue of reef corals. And within that context, my interest has always been on climate change, how they cope with various stresses in their environment, and in particular, what sort of photochemical mechanisms they use to cope. Now that I am in Alabama on the coast, my lab still does a fair bit of this work, but I've been really keen to get into coastal phytoplankton research as well, sort of have a local component to my research. And again, still focused on the same sort of general concepts, ecophysiology of marine algae and how they're coping or how these communities are changing in response to climate stress. And so within that context is how over the past couple of years I've been able to start various sort of local coastal phytoplankton projects. Being at the mouth of Mobile Bay is a particularly unique environment because it's at the end of a very large watershed, and so we have this huge influx of fresh water coming from upstate and as well as surrounding areas, and so that makes Mobile Bay and the surrounding areas a very unique area to work in. One of the more novel components of the Dauphin Island Sea Lab is the Alabama Real-Time Coastal Observing System, or ARCOS. And so this is a really great resource that has various buoys throughout the bay and also across the sort of local region, and allows us to track and monitor various physical components of the environment. And so this is a resource that's been led by Dr. Brian Zwonkowski throughout the past years, and it has recently been revamped by various ARCOS technicians and data architect Pat David. That's made it into this really nice user interface. If you go to the website, you can click on any one of these buoys, and it provides you with this nice screen of various physical properties of the air at that location, temperature, salinity, dissolved oxygen, and so forth. And so this has been really useful in terms of for researchers, but also for the local community as they engage with fishing, with various tourism-related activities in the area as well. One of the aspects that we don't have yet in Mobile Bay is biological monitoring, or any sort of consistent biological monitoring. And as Lisa showed in one of her figures, HAB monitoring has become increasingly more common throughout the coastal areas of our country. So lots of HAB monitoring on the East Coast, lots on the West Coast. We learned about the Toast Observatory in Texas, and then Florida has various observatories as well. There is a bit of a gap, or historically been a bit of a gap, in monitoring in the northern Gulf Coast, or specifically along the little Alabama coastline and into Mississippi. And so one of the components of a recent opportunity we've had was to fill this gap. And so fortunately, former Senator Richard Shelby allocated some state funds for us to start a harmful algal bloom monitoring program in Mobile Bay. And to do this, we were going to focus on using the imaging flow cytobots, which Lisa's showcased already. And sort of in general, sort of the pipeline for HAB monitoring here, we have our imaging flow cytoblots. They collect these great images of what's in the water column. These images are then run through a computer-aided image recognition software to identify what species they are, and through that system, we can have accurate HAB monitoring. And so this is very much the focus of our monitoring program in Mobile Bay, is to set up a very similar system for the Alabama coastline. However, part of the project that we're working on is to have sort of a broader network that encompasses monitoring, not just within the Gulf Coast, but throughout the country as well. To do this, a little bit of focus needs to be placed on the computer-aided species identification. So for as Lisa explained, to do this sort of monitoring, we really need to be able to classify the individual species. And to do that, you train a classifier in order to assess each individual image. And to be able to compare various data sets, ideally all these data sets need to be classified using the same system. That is the same image classification system that's been trained the same way. And so that's where various current projects differ a little in terms of their training. And that has to do with what kind of image recognition software is being utilized, what species are of particular interest, and what sort of resources has gone into data training. But to be able to monitor over large areas, you really need to be able to pass all the information through a central classifier system. And so that is something that GCOOS has been working on. Product developer Robert Currier is developing the Oceanographic Research IFCB Observing Network. And the idea here is to be able to automatically upload images from IFCBs throughout the region and have them processed through a central classifier system, and then have that resulting information available through the GCOOS portal for whichever end users are interested. This is still being developed, but the idea behind it would very much be a portal such as this, where you can directly go into an area of interest and select a particular IFCB monitoring system and look at the data output in real time, very similar to the TOAST system in Texas. As I mentioned, my group is very new to IFCBs, and so for support of this project, we're focused on cultivation of various HAB species so that we can image them and sort of assess or set up that initial classifier, or that is provide high quality images for the classifier to be constructed. And so right now we are cultivating various species for this purpose as well, and ideally also using images that have already been taken of various species of interest. Past providing the images for the classifier, we'll also be setting up an IFCB right off of our coast. So here we are starting on the right-hand side. Here we are at the very end of Mobile Bay. We have Dolphin Island, this little strip of land here, and right there on the very eastern part is the Dolphin Island Sea Lab. If you look closely at this Google image, we have a dock right here that extends a little into the bay, and that's actually where one of the ARCOS networks is currently located. And so not only is this an ideal place to deploy an IFCB because we're close to shore and can have shoreline power, run a hard line for data transfer and control, but we're also able to couple that IFCB data directly with some of the physical measurements that have been tracked there historically. All right, well, as I mentioned, this is a very new project to us. We got our first IFCB this past August, and our second IFCB in January. And so I have two PhD students, Jakayla Roseman Johnson and Shannon D'Alessandri, who are working on this particular project. And so they've recently this fall went to McLean Labs to get their certificates on running the IFCBs, and have been busy getting these up and running in our labs over the past semester. So that's essentially where we are with our HAB project. I'd like to particularly thank Darren Hendricks and Tuomo Sari for taking the time to meet with us over the past couple of months to discuss sort of best practices on how to deploy these instruments and sort of how allow us to learn from their experience in setting up the TOAST system in Texas. And that's about it. I do want to finish this up a little by discussing Shannon and Jakayla's projects and how the IFCB directly fits into their student projects. And so I was fortunate enough to get two grants funded by the Alabama Center of Excellence. And they're both basically focused on coastal phytoplankton communities and how those communities interact with important fisheries such as oysters. And so Shannon is looking at coastal phytoplankton within Mobile Bay and through our collaborations with Dr. Allison Robertson, we've been able to isolate single cells of various diatoms of interest. And the idea here is we can understand how they respond to various climate change stressors, such as ocean acidification, temperature, nutrients, all things that we're expecting to see change in Mobile Bay in the not too distant future. My other student, Jakayla, is taking this to the next step in understanding how these various phytoplankton species interact with oysters. Throughout Mobile Bay, there are a number of oyster aquaculture facilities, and understanding how the phytoplankton, what species of phytoplankton are consumed by oysters, and just as importantly, which ones are not consumed, could be really useful in understanding sort of where ideal spots are for oyster aquaculture and what conditions we need to be wary. And so for this project, she is running oyster clearance rate studies to understand which phytoplankton species are being pulled out of the water column, and then using environmental DNA to basically metabarcode feces and pseudofeces to therefore understand what species of phytoplankton are actually being consumed and what species are being filtered out by the oyster but then regurgitated or spit back out. That is, which ones are directly being selected for consumption versus being expelled back out. And so for both of these projects, there's a lot of good utility in incorporating the IFCB into some of the outcomes. Right now, we're using a combination of photobioreactor systems to keep cultures under very constrained environmental conditions, and so that's what you see here on the left. And what's novel about this particular photobioreactor are these black cables you see here, all of our fiber optic cables that allow us to monitor our cultures in real time using variable chlorophyll fluorescence so that we can understand their photobiology and their sort of ecophysiological responses to the environmental challenges that we give them. The Dauphin Island Sea Lab has recently set up an experimental system, having 30 individual experimental systems where we can modulate various environmental stressors such as ocean acidification, hypoxia, temperature, all in real time and having diel fluctuations. And so we're excited about using this to do some mesoscale experiments and looking at how coastal phytoplankton communities respond. And then I mentioned some of the oyster clearance rate assays that we've been working on to understand what species oysters are selecting for consumption versus regurgitating back out. And so our excitement with the IFCB, not only to monitor harmful algal blooms, but also sort of connect these experimental projects that my students are working on with direct measurements from Mobile Bay. All right, well that's all I have for you. Thank you very much.
Thanks, Kenny. That was great. I'm really excited to see you're not expanding just the network, but the workforce that goes along with it. A really important aspect of all this work. We will take questions. Do folks have questions? And Jen, if we can close down the presentation, I'll be able to see all of the participants in the hands. Thanks. Well, I have one while we're waiting for that, if it's okay. And this probably applies to both you and Lisa. You're both welcome to tag team on the answer. I'm curious, I know Lisa started working on this in 2007, 2008. I'm curious how long it took to get the buy-in from the stakeholders to use this as a decision-making tool. And where are you in that process as you're ramping up? Do you already have it? And are they standing by waiting to say we need this? Or are you going to have to make that sell to them to use?
Oh, that's a great question. I'm not sure I can answer that fully. I think Lisa might be better capable of that. But having said that, you know, the funding for this came from a state senator that recognized both the need for HAB monitoring in the region and the success of HAB monitoring systems elsewhere, like TOAST. And so there is some buy-in or some interest already I think it's up to us and use sort of the citizen science route of monitoring that's currently in Mobile Bay to sort of promote this as an elevation of what's currently available here.
I guess I can add to that we've had buy-in from the get-go, because in 2008 we had a major bloom and provided critical information to the Department of State or State Department of Health Services, Kirk Wiles. And so I don't think we've had any problem with buy-in of the stakeholders in that sense. Perhaps the oystermen might not like our work because it closes them down, but I think if they leave the oysters in the water and they depuriate the toxin, once the bloom is over, then they can still harvest them. They don't lose anything. They just may delay their harvest. So in that sense, I think we've had very good support from our stakeholders, not having to twist any arms or anything.
Thank you both for that. Other questions?
Well, I have a question. Are you aware of the Southern California Coastal Ocean Observing System? They also are trying to do a uniform or a pseudo global HAB identification program.
I was aware of the Southern California OOS, but I wasn't aware that they were trying to set up a
they have
network.
8 systems in there, and they're using the computer sciences group at Scripps to develop their. I don't know how far along they are, but rather than reinventing the wheel, you might want to talk to them.
Absolutely. I'm curious. One of the questions I'm thinking about there is how likely the same species would be found here versus on the California coast, and so thinking about some of the classifiers and how data would be compiled for both, if they can all be kind of sent down the same pipeline, or if they'd have to be some manipulation occurring.
Yeah, I would be concerned about using the same pipeline for every location, because you're going to have different species, and you're going to have the confusion matrix is going to be different with different community compositions. So I'd be a little wary of doing that.
And Kenny, we have a question from Mina asking, how often does Mobile Bay experience harmful algal blooms?
That's a good question. Major harmful algal blooms, such as those that are frequent on the Florida coast and in Texas, might not occur as often, but we do have small blooms along the coastline from time to time. I think there was a large one in 2016 that was absolutely viewable from afar. Admittedly, I haven't been here long enough to have much of a historical context, though.
Okay, and Greg put a comment in that Clarissa Anderson from GCOOS would be the contact, and we're happy to put you in contact and make that connection for you if desired.
Awesome. Absolutely.
Thanks for that comment, Greg. Anybody else? Any other questions, comments? All right, well, we're coming up on the hour. I'd like to thank our speakers, Lisa and Kenny, and it was really informative and great talks today, so really appreciate your time. I know how busy everybody is. I'd like to thank Jennifer Vreeland for all the technical support throughout the webinar series, communication support from Nadine Slimak, and to all of you for joining us throughout this series. On behalf of GCOOS, we thank all of our investigators for the important work that you do to provide information about the Gulf's coastal and open waters, and that benefits people, ecosystems, and the economy. We appreciate all that you do. Thanks for joining.